students, you are studying chapter 2 sexual reproduction in flowering plants with me Jyoti Tiwari. And in previous lecture video we have learned about ovule structure as well as female reproductive organ in detail. In this lecture video I am going to tell you about the development of megaspore as well as female gametophyte. This process is called megasporogenesis as well as macrogametogenesis. What is the difference between megasporogenesis and macrogametogenesis? Megasporogenesis is a process in which there is formation of mother spore cell is forming in megasporogenesis, and in macrogametogenesis there is formation of female gametophyte takes place. Let's understand it in detail with the help of diagram as we do. I am going to zoom the diagram. So I hope you are capable to visualize the diagram. This is megaspore mother cell. It is formed by megasporogenesis process. Okay, and this megaspore mother cell undergo division of meiosis. Meiosis is such type of division which is called reductional division. I have explained you it previously. Please watch the previous lecture video for more detail. And so here four type of haploid cells are forming. Means here chromosome number is reduced to n. These four haploid cells, out of which 3 haploid cell get degenerated, and only there is one cell which is going to be further in development, get nourished by the nucleus. This functional megaspore undergo division. It perform function. It get nourishment from nucleus and grow in size. This functional megaspore after that localized in the micropyle region, which now this megaspore after getting nourished further divide by meiosis. Meiosis is not a reductional division. In this chromosome number not going to be half. Means here the nucleus is going to be divided into two nucleus are formed. After that these two nuclei again undergo meiosis division, and this nuclei formed divided into 2, and this nuclei again divided into 2. This is the 4 nuclei stage. This is called 4 nuclei stage. After that this nucleus again undergo meiosis division. You can also look at it in this diagram. Suppose this nuclei going to be divided and form 2 nuclei, and this nuclei again going to be divided into 2 nuclei. Again this nucleus going to be divided into 2, and this again divided into 2. Means 4 nuclei are here, 4 nuclei are here. Out of these nuclei, 2 nuclei get localized in the central region. From opposite pole 2 nuclei move and localize in the central region. These nuclei further fuse. Their nucleus does not fuse, only their cytoplasm fuse. For example, this and this going to be fused like this manner. Only their cytoplasm is fusing here. Nucleus does not undergo fusion. So this is the 2 polar nuclei, and remaining three nuclei in opposite pole called antipodal cells, and remaining these 3, out of these 3 these nuclei, 2 nuclei form synergid. 2 nuclei form synergid, and 1 nuclei form egg cell. 1 nuclei form egg cell. You can also see it here as 2 is going to be formed. These are synergid. These are synergid, and remaining 1 form egg cell. Remaining 1 form egg cell. So again, come back to this diagram. These are called antipodal cells, and these are called synergid cell, and this is called egg cell. Again, it undergo fusion and called secondary nucleus, formed by fusion of 2 polar nuclei. Means here 2 polar nuclei are going to be fused. Here it is written by little bit a mistake, because it's in printed form. Actually there is not nuclei is fusing, only cytoplasm is going to be fused. Okay. After that, you can see such type of structure in the ovule. This is the structure of ovule, and this located in the embryo sac. Embryo sac located in this region. Embryo sac located in this region. I have previously explained the diagram of ovule in detail. Please watch that lecture video to understand it here. Okay, it's very necessary. So this is the structure of young ovule. What are the function of these antipodal cell, polar nuclei, egg and synergid? Let's understand it. So the function of antipodal cell is nutrition. It provides nutrition to the developing zygote after fertilization. And polar nuclei function is in secondary fertilization. It take part in secondary fertilization. Egg cell, it also take part in fertilization, fuse with the male nuclei which is coming from pollen tube. Synergid help in fertilization. It help in fertilization. How it help? Let's understand. There is the filiform apparatus present here. Filiform apparatus present here, which actually help to continue with the pollen tube, from which male nuclei is coming, and male nuclei after coming fuse with the egg cell. Okay, so in this way fertilization takes place, and synergid cell is helping egg cell to fuse with the male nuclei, because there is filiform apparatus is present. This is also called as egg apparatus. This hole in this end is called egg apparatus. Okay, so I hope you can understand what I am saying, and I will continue in next lecture video. Thank you for watching.